thing I'm talking about uses principal components analysis. So this is probably the most famous use of principal components analysis on genomic data. So just to set things up, to make this pretty picture, what John Novelli did was he took genomic data from about 1,300 European humans and found that when you place each of those samples on the major axes of genetic variation from those genomes and color them according to what country those individuals were from, you nicely sort of recapitulate the map of Europe. Okay, so that's genetic similarity, recapitulating geographic proximity, which often happens using PCA, and is partly why it's useful. Here's another nice example showing sort of how different land races of maize are related to each other and to the wild ancestors. Okay, so in general, what we use PCA for is visualizing population structure. You know, how different populations are related to each other vaguely. Okay, so before I get into it further, I want to step back a little bit to think about what's going on, because it's not very obvious. And it might not be any better after I say this, but so principal components, what it's doing is it's describing major structure in the genetic covariance matrix. And that's somehow related to genetic similarity, and maybe more concretely, you can think about it as follows. So to compute the genetic covariance between two samples, I and J, you can do it as follows, roughly. So first pick two other samples at random from your data set, and then you're going to average over those. Those will be X and Y. And then look across all the loci where there's mutations that separate your 4 samples into 2 groups of 2. And every time you've got a mutation that puts I and J, the ones that you're computing the covariance between together, you add 1 to your score. And every time you get a mutation that puts I together with I, separate from J and X, you subtract 1. So you sort of average that across X and Y and also loci, and that gets you the covariance matrix. Okay, so if I and J are closely related in many parts of the genome, then that will happen a lot, and they'll have higher covariance. Okay, so this is, see here I'm drawing something related to the local genealogical trees along the genome, and so this is somehow summarizing those trees, so the average patterns of relationships along the genome. Okay, so that's somehow in an inscrutable way maybe summarizing population structure. And so, well, okay, what is the population structure that we're trying to summarize? I believe what we mean is that's something to do with historical patterns of interbreeding, migration, population size changes, that sort of thing, demography. Okay, but like we all know that, I guess, we don't actually get to infer the demography, the demographic history, we just get to infer, the trees, and not even then, it's the mutations on the trees, right? So selection can have a lot to do with that. We all know that selection can locally distort, the genealogical patterns moving along the genome, specifically link selection. So sort of a fairly obvious example is local adaptation. If there's a region of the genome with a bunch of locally adapted alleles, that'll select against migrant alleles in that part of the genome, and so the local trees in that part of the genome will be more structured in space, in principle. And there's lots of other ways that link selection sort of distorts these genealogies locally. Like common example is the dip in diversity around non-synonymous substitutions. What this plot here is saying is that if you look at a non-synonymous substitution in between 2 Drosophila melanogaster, the mean time to common ancestor is like 5 or 10% less than in other places on the genome. Okay. Okay, so this local distortions of genealogies by selection, this is just what we use to do scans for selection, right? Like FSG scans or whatever, right? And So when we get Manhattan plots, these nice big skyscrapers are like very small, strong distortions of the genealogies that make the local patterns of relatedness in that little tiny chunk around the gene under selection, hopefully little tiny so we know what it is, make the local patterns of relatedness look very different than sort of the rest of the genome that we like to imagine as, all following the general patterns of relatedness set up by population structure. Okay, but we also know that link selection can have much broader effects than this. There's been a lot of studies showing sort of larger scale correlations in various patterns of diversity, probably because of like background selection and other sorts of link selection. So I kind of went into this, my student went into this wondering like, well, how strong might that effect be and what might it tell us? Is there a significant, these are little windows of isolated, really weird patterns of relatedness, but is there sort of larger scale shared variation on the chromosome scale? All right, so that's the motivation. So to have a look at this is what we did. You take a bunch of samples and you chunk up your chromosomes into chunks. And then in each genome window, you do PCA, sort of independently on each window. And then what you want to do is you want to use the PCA to measure the similarity between different parts of the genome, the similarity in patterns of relatedness between different parts of the genome. Okay, so the way to sort of think about it is you take two windows and you look at their PCA plots, and then you measure the similarity in those pictures up to rotations and scalings and flips and things. That's not actually how we measure the similarity, but it's close enough. Okay, so from this, you can get the distance matrix that tells you how similar any two windows along the chromosome are to each other in terms of patterns of relatedness between these samples. Okay, and that's still not a nice thing to look at, so to visualize what's going on, we then apply MDS, multidimensional scaling, to that distance matrix. So you get things like this. This is here, position along the chromosome. This here is MDS coordinate 1, so points on this figure are windows along the genome, and if 2 points have similar y coordinates, that means their local patterns, their patterns of relatedness, their PC plots look similar. Okay, so on this chromosome, clearly something's going on, and then you might want to say what? Like there's significant variation in patterns of relatedness along this chromosome, so to kind of see what's going on, what we do is you can pull out a subset of windows, for instance, the orange ones that are most extreme in this direction on this axis. Pull them out, you squish them together, then you do PCA on that collection of windows to visualize what population structure looks like there. So then you do it separately on maybe the green ones and the purple ones too, and compare them, and then hopefully it makes sense. Okay, so that's the method. We have it written into an R package. It's here. There's also our markdown templates and some command line scripts with like documentation and stuff. So it should be pretty easy for you to try it on your data if you want. And so you should try and then tell me what happens, because I like looking at pretty pictures like this. Okay. But first, let me show you what happens on a couple of data sets. First, we applied it to the DPGP. This is a few hundred Drosophila melanogaster from mostly across Africa. So I was trying to pick data sets with whole genome sequence so that we have really good resolution and can look for variation along genomes, and also good spatial sampling, because that's the thing that's setting up the patterns that we expect selection to be distorting. Okay, so then if we apply the method to that data set, what we see are these well-known large segregating inversions, in retrospect, unsurprisingly. So this is chromosome 2R. These lines are the breakpoints of that inversion. You see the main sort of variation in population structure along the chromosome is like inside versus outside of the inversion. And then if you pull out like these green windows, the ones that show most extreme population structure in this direction, and do PCA on them, it tells you, so this is the PC plot, so here points are flies, and they're colored according to which orientation of the inversion they have. So it tells you who has what orientation of the inversion. Okay, so it's kind of a good exercise to figure out why the inversion pops up so strongly, but instead of talking through that, I'll show you what happens when you remove the inversions. So we marked as missing all the haplotypes that are in the the less common orientation of each inversion. Or I should have mentioned that all the chromosome arms look like this, because they all have these big segregating inversions. Okay, so here's what happens if you take those out. So this is now all the chromosome arms. And on the top is along the chromosome against MDS1, so this sort of measure of patterns of variation, and they're all on the same axis. These are comparable. And then down here, we've got recombination rate. So you see as there's these nice correlations along the genome, correlated with distance from centromere or something, but also correlated with recombination rate, which is a good proxy for strength of link selection. Okay, so my explanation is this is some kind of link selection. And we also applied it to the Medicago Trincatula HapMap project, which is also a couple hundred whole genome sequences, this time from around the Mediterranean. And here you get the same thing, except for with no inversions. On the bottom here, instead of recombination rate, is gene density. So the data were better here for that. But it's also a measure of strength of local selection. The local strength of link selection. Okay, so at this point I would like to cut out all the windows that are up here and do PCA and down there and show you the differences in the PCAs. They're pretty substantial. And then explain to you what kind of selection is causing the patterns that we see, but I don't know how to do that. It's too confusing. I mean, there's probably lots of different things going on, and right, so the point of this is to provide a method to visualize these patterns and to show that there is this sort of substantial structure along chromosomes. So in case you're wondering, you could probably think of various other things that aren't related to link selection that could be causing this, like missing data or recombination rate variation for uninteresting reasons, but it's not those things. I'm happy to talk about those later. Okay, so sort of in conclusion, some more strong evidence for link selection, and also I guess a point coming out of this is that when we do things like GWAS, we control for population structure using the kinship matrix, and this implies that maybe there's kind of more than one kinship matrix we should be using. Okay, thanks to Han, you did a good work.